Okay, so let's go ahead and get started. I had someone email me, thank you. If ever anyone finds a broken link, please let me know because I might not be aware of it. All right. So under the respiratory chapter, because next Friday is our quiz, our lecture quiz, it is going to be just like last time, 25 questions, Scantron, so bring a pencil, all right? Why can't I get this thing out the way? Here we go. And so what was not working was the study outline. It basically was a broken link. So it's working now. All right. And I double checked that everything, the practice exam is showing up. So that's good. And for that particular chapter, let's get back to where we were. I covered a lot of stuff this semester. Holy cow. We also have instructor notes. Now, those have not been updated recently, so they might cover some material that I no longer cover in class. Don't worry about that because I do change things and update them and add material that's current. So just worry about the study outline, the practice exam for that chapter. Everything else should be working, but should you encounter a link that's not working, please let me know. I might not know it. And also for the kidney physiology, it originally said chapter 11, but it did pull up the right kidney stuff. So I don't know where my brain was, but I fixed that. So everything's correct now. And also I picked up the donation boxes yesterday, so they're gone because today I'm bringing them to the center. So if you donated, it's going to them today, and that's going to really help some animals. Okay. All right. So any questions about any of that? Because we got to finish immune physiology. Okay, so we left off with how you can study this chapter. It's really, waiting for this, here we go. And boom. If you want to study this stuff, like the sequence, because I'm going to ask this question on an exam, from when bacteria enter into your skin, through your skin, to antibodies are produced and what they do, go over that. And then if you want to look at the categories of the immune system from innate and specific or adaptive, we broke all that down. Obviously, within the notes, there is more information about each of these cells here. I couldn't fit it all on this little flow diagram, but consider these vocabs to know what complement proteins do, know what mast cells do, natural killer cells, what an APC is and what it does, and so on, so forth, up that. And then, of course, same thing here, know what they do. So there's more to this outline, but at least you can kind of break it down in your brain easily with this flow diagram. Okay, so we basically have a couple more slides of material that I could ask on an exam. And then the rest of this chapter is actually an educational thing for you as future nurses, because what you're going to encounter is, well, with many things, a lot of misinformation and hearsay and fears that people have that really aren't founded on anything. And I kind of want to give you some information so that when you're talking to your patient and you're working on patient compliance, which is they follow the recommended guidelines of their physician, okay? You're going to be spending more time talking to them. Okay, so classification of immunity, two classifications, active, passive. If it's active, your immune system has to do the work of making the antibodies. And we saw that sequence of events that happens, bacteria or a virus enters your body's never seen it before, and it takes 2 to 3 weeks to get to antibody production, and then what antibodies do when they encounter the pathogen for which they were made. All right, that takes work, 2 to 3 weeks of work. All right, that's why it's active. And we've got two subdivisions of that. And passive, your immune system does not have to do the work at all. You're given the antibodies. All right, so two classifications of that. Active immunity, you have to do the work, subdivided into active natural immunity. So let's see what my pen is doing this morning. You have a natural exposure. And make antibodies. This is how a lot of our immune system functions. We're exposed to something, and then our body goes through that process 2 to 3 weeks. Your innate immunity is protecting you in the meantime. And then once antibodies are formed, hopefully you're good for a while. But it is active. Your body has to do the work. The other form of active immunity is active artificial. Any ideas what this would be? Your body does the work to make antibodies, but what was your exposure? Vaccination. All right. You are vaccinated and you make antibodies. Or you could call it immunization, whatever you want to call it. But your body has to do the work. Now, if it's passive, they're given to you. So if I have up here under passive natural, what would be an example of? Well, it's right there, right? What does that indicate? Passive natural immunity. You're getting the antibodies from breast milk. All right. So a woman who breastfeeds her child, any antibodies that her body has made up until that point in her life, those antibodies pass across into the milk and the baby gets it. All right. So that is breastfeeding. One of the many benefits of breastfeeding is, well, when the baby's born, they come out, they're not fully cooked. I just like to say that because it just seems to make sense to me. Their immune system is not ready to function yet. It's too immature. It's going to take some time. For people who are in the know, what is the earliest time that a baby can be vaccinated for, say, measles, mumps, rubella? Anyone? Something like 2 months. All right. Because it takes about that time for their immune system to be able to respond and make antibodies. Before that point, there's no point in vaccinating them because their immune system, like, I don't know what you want me to do. I can't do anything about this. All right. So there's no point in vaccinating them because they can't mount a response and make antibodies. All right. Same thing with dogs, cats, any animals. Once your immune system is active, then vaccinate. When it's not active, don't waste the time and money. Okay. And then there is, oops, that's in the way. Of course it is. Passive artificial immunity. So what's this indicating? Have any ideas? That's a tougher one. I'll give you a for example. So I just had someone from that Black Creek Wildlife Center come this Tuesday and give a talk on reptiles. The reptiles we have in South Carolina. And of course, he started talking about what makes sense with the warm weather coming, venomous snakes. And he's talking about how do you tell venomous from a non-venomous? And they said, what happens if you get bit, And so he's talking about, doctors got to have anti-venom on hand or else you might be in big trouble. So what that is, you get injected with antibodies that have been made against that venom antigen, that venom toxin. The antibodies bind to it, inactivate it, and save your life. Tetanus antitoxin. So you have to be tetanus vaccinated to be a student. Has to be kept up every 10 years at least. If you get a deep puncture wound, you're supposed to go in, get a booster of the tetanus vaccine just to make sure you're good. Now, if your vaccine is like mine, I got to get boosted. If it's been 10 years and you get a deep puncture wound, yeah, they will vaccinate you at the hospital, but they'll also give you an injection of basically antibodies that are going to defeat the tetanus toxin in your body so that while your immune system is prepping up again to make antibodies again or to top them off, the injection of the serum of antibodies is protecting you. So you're going to see this in nursing. All right. So you are injected, an injection of antibodies. So that's that. Okay. I remember one time I had a billy goat and he was a big billy goat and we had him separate from the girls and the girls were teasing him and waving their little tails and bleating at him. He couldn't stand it. And he tried to scale our fence and we had some like wire, not barbed wire, but we had just, nothing could poke through the other side. And trying to scale that fence, he got a deep cut and he was fine. The vet came out and she gave him a vaccine of the tetanus and she gave him the tetanus antitoxin just to be on the safe side. He was absolutely fine. So this happens for people too. Okay, autoimmune disorders, ones that we have talked about. Actually, we didn't talk about rheumatoid arthritis. So I'll just cross that off. But that is something that you should be aware of in nursing. Rheumatoid arthritis, young people can get it, all right? It has no bias towards age. And your immune system attacks your joints and you get a chronic inflammation. And what happens to tissue that's chronically inflamed? it breaks apart. Scar tissue forms, and that scar tissue becomes stiff and inflexible, and you have stiff inflexible joints that are painful, and left untreated, the inflammation will eventually eat away at the cartilage and it's gone. So you'll see people come in, could be just old age arthritis, or it could be this where immune system's attacking it. But we've talked about rheumatic heart disease, antibodies that your body produces attacks heart valves, particularly the bicuspid valve. All right, so that's between the left atrium and left ventricle. Multiple sclerosis, we covered way back, where your immune system produces antibodies that break down myelin on your neurons. We talked about Graves' disease, which is an autoimmune attack on your thyroid gland, causing it to enlarge, and other symptoms like that. And then, of course, you're all very familiar with that, so I don't have to talk anything more about myasthenia gravis. All right, we've got that one down very thoroughly. So technically, oops, I went too far. That is the end of what I'm going to ask on a test. I'm going to add the term herd immunity. I put it in red because it's not on the copy of the PowerPoint that you have. So this is where you can just listen, ask questions, all right? Because the material that I'm expecting you to know is done. This is more like, I'd like you to know the history of this so that you can be educated for your patient's sakes and be an advocate for your patient and their health, because this is very important. I don't have to stress this much more because we've lived now through COVID. All right. We've lost many due to COVID. And there's still pockets of resistance to vaccination. It's become political. I don't know why, but the health community pretty much agrees that getting vaccinated for this is important. And as future nurses, you're going to have to be vaccinated if you aren't, because if you want to do your clinical rounds at McLeod, or if they send you to MUSC, I know MUSC, or I might have this backwards, one of the two, requires that any nursing student be vaccinated or they can't enter the building. So you can talk to the nursing department about that. But I did talk to Dory Weaver and I asked her that specifically because I wanted to know what their policy was, and that's what their policy was. So if you had this in microbiology, great. I know Dr. Pryor covers it, the history of vaccinations, but vaccinations went on way centuries ago. It was more like a natural vaccination, natural exposure. But he noticed that milkmaids who would milk the cows for whoever owned the property rarely had smallpox. Now, you've probably never heard of smallpox because you don't worry about it now, because we had a vaccination that basically wiped it out, put it into inexistence. Basically, there's no good host for smallpox right now. So he thought that maybe because the milkmaids were milking cows and cows have cowpox, sometimes they have those lesions on their udder, the milkmaids were exposed, and the cowpox was similar enough in viral structure that the milkmaids produced antibodies that were also good against human smallpox, which was incredibly deadly. In Europe, smallpox had been around for a while, so a lot of people had natural exposure. But when they came over and they settled the Americas, Native American populations were virtually eradicated because of smallpox. And there's history that they deliberately gave smallpox infected towels, and not towels, but blankets to them to basically help wipe out the population. Well, anyway, so you can't do what he did now because you can't experiment on humans unless they can consent, and a little boy can't consent. But he inoculated a boy with cowpox. And then I don't know how much time he waited. I don't know this. I didn't read it. I just have a summary. But then he exposed him to smallpox, human smallpox, which could have killed him. But he apparently had the antibodies from the cowpox and it saved him. Now, this is the story. How many kids did he actually do this with that died before? He got one kid that made it. I don't know. But anyway, so that's our first vaccination. But it's a natural exposure, not something that's made in a laboratory, which is what we do now. You're not going to advance for me? Okay. We'll do it this way then. Okay. It's frozen. I don't know why it froze up on me. Don't worry. I've got a solution. Escape. Keep. We will just make it. This thing is such a pain. We'll just get it going again. It's Friday. I have to have some sort of technology issue. Okay. So the importance of the vaccination, this is what I want to try to impart onto you. Why do we get a series of vaccinations? Little kids get usually 2 or 3 series of measles, mumps, rubella. We had a series of vaccinations with COVID. Usually flu is 1 vaccine is good enough. But here's what we have. When you have your primary response, the first time that you're exposed to something, whether it's a natural exposure or you've been vaccinated, here's what happens. So I made it blue, so the blue line. So you're exposed, and notice week 1, week 2, week 3, this is plasma antibody concentration. So it takes some time for you to get that peak antibody titer. And in the meantime, your innate immunity is hopefully protecting you. All right, so that's a pretty good amount of antibodies after 3 weeks, okay? And you get memory B cells that are produced and things like that. Okay, the next, so here's, I'm the Lord of the Rings geek. Here we have the little, hobbits, and here is the pathogen coming in on them. Their first response to defend themselves is okay, but it's so wrong. All right, that does not put the fear of anything into the pathogen that's coming to kill them. When you get your second vaccine, okay, immediately upon exposure to that second vaccine, boom, it goes up, it maxes out. Because at the first time that you produced your plasma B cells, and when they became exposed to that antigen again with the vaccine, they threw their antibodies at it. There was agglutination, and then there was clonal replication of more plasma B cells and memory B cells. All right. So when you have the second exposure, you have basically an army of B cells that are going to protect you. So that second vaccination was pretty important because the scale of the defense is much higher. And that's why they do this series of vaccines. So it's important when you have people coming in for their vaccine, whatever it is, especially little kids, it's important. You must come back at this time to get the booster, because what you're hoping to do is this right here. And that's why we had, if you had the Pfizer or Moderna COVID vaccine, 3 weeks after your first vaccination, you had to get a second one. And this is why a lot of us are not having to wear masks anymore, because we've got this level of protection and we don't have enough hosts that are unimmunized to serve as a vector for the virus to replicate and spread. We're reducing the number of human bodies that the virus can infect. And so you're cutting the virus down. Viruses can't reproduce on their own. They need our cells. If our cells are protected, they've got nothing to reproduce us. Guess what? The virus can actually go extinct. And we have proof of this. All right. Or to be at such low levels in our population that it's virtually undetectable. So measles, mumps, rubella. Let me move this stupid thing again. Okay. Before the measles vaccine came into existence, a lot of kids who are vulnerable because their immune system's not quite competent yet, it's not very good, they would be scarred forever, or they would actually die. But a lot of kids came down with it. It just happened. Adults with a more competent and experienced immune system, less vulnerable. So who needs vaccinations? We all do, especially if it's a very bad virus or something, but definitely kids and elderly people whose immune system's kind of weakening. So a lot of cases with 500 deaths per year, and I think that's extremely conservative. After the vaccine, 99.9% decrease in the incidence of measles, mumps, and rubella. Okay. In fact, we had it almost eradicated until rather recently. It's frozen again. So what I'm going to do, because it doesn't like this mode, so what I'm going to do, what I did before, and just come back, escape out. And I'm just going to keep it like this. I think you can still see it. All right. If I do it any other way, it's going to freeze up on me. So it's going to be small. You don't have to know numbers. So basically measles, mumps, rubella, I put it all in blue. Pre-vaccine era estimated morbidity. People were infected, they were showing symptoms, especially children. It was really bad. So those numbers are really high. After the vaccine, almost no cases. After the whole population was well vaccinated and had herd immunity. You probably don't even think about it, right? Do you know of anyone who's recently come down with measles, mumps, or rubella? There's pockets in the United States that are called the anti-vaxxers, and they held on to this one study that one scientist did that was extremely flawed, and they believed him. And then also someone started, I forget her name. She dated Jim Carrey. What was her name? Anyway, she was a very public persona, and she started saying that vaccines cause autism with absolutely no proof of that. And people bought into that, and they said, well, I'm not vaccinating my child. And guess what? Children were coming down with measles again. And hospitals have to be like, is your kid vaccinated? No, you can't come into the hospital. We've got very vulnerable children and people here. You can't bring that in our hospital. Get vaccinated. You don't think about polio. Do you even know what polio is? Some of you might have learned about it. You don't worry about polio, do you? Vaccination basically eradicated it. There's no hosts anymore. All right. Smallpox, again, 100% effectiveness of that vaccine. You don't worry about getting smallpox, do you? Virtually eradicated because almost all the adult population we have, especially in the United States or the Western world, the virus got eradicated because the vaccine made no new hosts, and without hosts, the virus can't replicate. And if it can't reproduce itself, it goes extinct, or at such low levels that you don't worry about it. So do vaccines cause autism? No. Honestly, what probably is going on, people are saying there's more cases of autism. We're getting better at diagnosing it earlier and earlier because we're learning more about it. I think in the past, like 100 years ago, they simply didn't know what was wrong with these people. And so they couldn't put a label on it. If you can't put a label on it, you can't work on treating it. But there's no definitive proof that it causes autism. So here's the controversy. So back in 1995, pre-you, okay, he published an article in The Lancet, which is a good journal. His name was Andrew Wakefield, and he had a sample size of 12 students. Have you noticed that sometimes in our lab, when we have 12 or 13 people in lab and we're putting our class data into the computer, and sometimes I have to tweak the data because it's skewed, it's got experimental error because the sample size is small. I've mentioned about small sample size before. To draw big conclusions from a sample size of 12 is just not done. You're not, I wouldn't be robust. I wouldn't say the data I got from my physiology class of students of 13 people, I can make broad general statements for the health of everyone in the country. No, I wouldn't trust that data at all because the sample size is way too small and you can't rule out all the variation that can happen. But he basically had 12 students that had the 3 measles, mumps, rubella vaccine taken together. And this is a very different schedule at that time than we have now. And he said these very vague statements could alter immune systems. Well, yeah, it's making your immune system produce antibodies to the thing you just injected them with, cause intestinal woes, which I don't even know what that is, and then could damage the brain. He had no empirical data at all. It was just suggested. It was just, he just posed these ideas. But the problem is, anytime you do research, and I know this because I was in grad school and I did research and published my findings that had to be reviewed by a group of scientists who criticized it, said, no, you need to fix this, send it back to us and we'll think about publishing. And it took about 1.5 years just to get one article published. It's scrutinized. And then what happens, and I've had this happen, I've had someone, so I keep on these publication websites, and they let you know who cited your article and whatnot. And this, and if they publish something citing you, or if someone repeats your study, it lets you know. And I actually had someone repeat my master's work, and thank God they got the same results. Okay, I was like, whew, okay, my data was good, it was robust. But when the scientific community tried to repeat his study, they couldn't find it. It's just, they said, no, we don't find that at all. So that puts big doubt into the scientific community when someone replicates your study and they don't find what you find. So they did say he had sample bias. It's too small of sample size to draw any reasonable conclusions. The medical journal called his research fraudulent. I would about die. I think I'd go to, where would I go? New Zealand and hide out for the rest of my life. If the scientific community had called my research fraudulent and said, this is a bunch of crap, or studies crap, I would just about die. My integrity as a scientist would be, whew. Okay. The British journal Lancet retracted his publication. They took it back. All right. And the medical authorities stripped him of his medical license. That's how bad it was. So he really got punished for it because it was crap. The problem is people still believed him, and it was used to promote anti-vaccination rhetoric. And we do have pockets in the country where, from not vaccinating, Measles is showing up again. And have you heard, you'd have to live under a rock to not hear about the pockets of the population who didn't want to get COVID vaccinated, right? For whatever reason. My rights, my body, my choice, right? Well, not really, because your body could be a vector for viral replication. And then you walk around without a mask, not vaccinated, and you come in contact with a patient who's taking autoimmune immunosuppressants because they have myasthenia gravis and they're trying to slow the progress of their disease, but their immune system is immunosuppressed. And you come in contact with them and you expose them to COVID that you're carrying asymptomatically. And then that person is really sick, possibly could die. We vaccinate ourselves to protect ourselves, but if we are really healthy, even if you got COVID, you probably weren't hospitalized and intubated. The people that you're protecting are the very young children whose immune systems aren't competent yet. People who in the population who have suppressed immunity because they have a disease, they're battling with other health concerns, high blood pressure, diabetes, all kinds of problems, which makes them overall weak. AIDS patients who are immunosuppressed, and older people who are battling health problems and their immune system is not as strong as it once was. That's who you're protecting, because you are not harboring and replicating the virus in your cells, walking around asymptomatically, but you're still carrying it. You're still a vector. And then you come in contact with those people and you expose them, and they're the ones that suffer. That's why we vaccinate. Okay, so I sound like I'm on some sort of, I'm literally on the podium preaching, but it is important as future healthcare workers because you're going to come across this. And hopefully you'll be able to explain it to them in a nice way, because what you would like is patient compliance. Now you're going to have the stubborn people who say, screw you, right? But then that comes at a cost. And sometimes in certain doctor's office, they have every right to refuse you service because you're not vaccinated, because they don't want you coming in asymptomatic carrier and then exposing all their patients who might be vulnerable. They could be very well turned away. So anyway, herd immunity is what I just described to you. When enough people in the population are vaccinated, there is no reservoir, human body reservoir for the virus to live in, replicate itself, and spread. You're denying the virus an ability to reproduce itself and spread. And that's what herd immunity is. So vaccination, and you might have read about this because we were learning a lot about COVID. Like a 50% compliance in vaccination is really mediocre, but we'll take that. Actually, looking at a 90+% vaccination rate to really provide herd immunity. And that's kind of what the goal is to have that high level. So 95% is what medical doctors and epidemiologists and virologists agree. That's a good vaccination percent for a community to protect that community. Now, so here we have healthy person in blue, but they're not vaccinated. And then we have an immunized person in yellow, and we don't have any immunized people in this population right now. And we have the sick people. Okay, so the red is the COVID people. I just use it because it's a good example. Over time, you have two infected people. They're not wearing masks. They're not vaccinated. They're out in public doing all the things we were told not to do. And then all unimmunized but healthy people, after a time, that whole community becomes infected. And that's what we were trying to do with COVID, is try and stay home, wear masks, wash your hands, try to not go out in very public places if you can help it. A lot of events were shut down, and the university was the same. We went completely virtual education. We hated it, but we didn't want this because we didn't know what we were dealing with. COVID was new to us. All right, so we were scrambling to come up with a vaccine, scrambling at how deadly is this? Oh my God, you know? Okay, I know what happened. See, without doing it in regular PowerPoint. Okay, so now the next thing, the yellow people are immunized. The infected people are there, and the blue are the healthy at this time, but unvaccinated. Notice the blue people that had a yellow person in front of them, okay, remain uninfected, all right? The people who are vaccinated are literally a viral barrier to unimmunized people. If the person that you're working around or interacting with is vaccinated and you're not, they're vaccinated. Their immune system defeats the virus if they've been exposed. So they're not reproducing viruses in their cells and then becoming infective. They're protected. Now, I do know that vaccinated people can still come down with COVID, and the ones that do tend to have very mild symptoms, and that's possible. But getting vaccinated is better than not being vaccinated. So some people in the community where they're surrounded by more vaccinated people are protected. And then down here, when you have a high vaccination rate in the community and you have 2 infected people and we have unvaccinated but healthy people, you have less infection going on because you're surrounded by people who are vaccinated. So imagine these blue people were children. Imagine these blue people were people taking immunosuppressant drugs or had immunosuppression from a disease they're battling or other things that made them extremely vulnerable. They're protected because vaccinated people are the ones around them, not the unvaccinated sick people. So that's herd immunity. Does that explain all those things pretty well for you? And this was just showing how outbreak happens when you don't have very many yellow people that are vaccinated. And they're just basically saying, shooting for that 90% or above, the virus has got nowhere to go and reproduce because everybody it encounters, it can't infect very well before that person's immune system defeats it. And so the virus has got no way to reproduce. If something has no way to reproduce, over time, it will go extinct. It just can't replicate itself. And if the people are immunized and they don't act as a reservoir for the virus, not only can the virus not replicate itself, if it mutates, people who go unvaccinated, they get infected with the virus, and as the virus is replicating itself, it's mutating into a new strain. You've heard about that, right? People who are vaccinated, if you have more vaccinated, the virus has no place to replicate and it has less chance to mutate into another strain that produces a problem. Okay, so that's a great way to end the immune system. And it's a little after 10. That's too early to break, but I will post these notes after class, and anything that was after that last review slide, I'm not going to ask you except for basically the definition of herd immunity. All that other stuff I talked about was something that the medical community is pretty ardent about. And I think as future nurses, you should know the story, where it came from, and then what the importance of vaccination is, getting that second booster and why it's such a good protection. And then, of course, the concept of herd immunity. Okay. Did I have the reproductive PowerPoint here? Okay. I got to find it. I guess I didn't have it here. Let me find my PowerPoint. Okay. Okay, and when did I modify it? There we go. So reproductive physiology. Wake up. There we go. Now, I don't know if I try and make this one go full size. Let's just see what happens. So the first slides are going to be review. You cover reproductive anatomy in anatomy, right? Hopefully. I knew of a time when they didn't cover it, and I was like, really? This is a topic that your patients are going to come in, and some people are really forthright and comfortable about their bodies and talking about sex, sexual function, sexual anatomy, and what's going on. And others are going to be like, I gotta, and they won't use the actual terms vagina, penis, anything like that. They'll use some euphemism because they've never been made to feel comfortable talking about those things. So you as the nurse have to be the one who's comfortable talking about this. So if you can't get over the giggles and the laughs, and your patient is not, you have to make them feel comfortable in your presence, because what you want is your patient to actually be truthful and honest with you and feel that it can tell you things, private things about them. So yeah. All right. And remember, I'm a reproductive physiologist, so this is my thing. So review. You should know, of course, that this is the scrotal sac carrying the testes, and then this testes is where the sperm are produced, and the sperm transport tubes include, well, this epididymis, which is like a little comma that sits like over the testis, stores sperm until it's mature. And then during an ejaculatory event, it goes up the vas deferens, which does not go through the bladder. It goes around the outside of the bladder. And then there's a seminal vesicle here that produces the liquid component of semen. We'll talk about that. And then another transport tube that this one does go through the prostate is called the ejaculatory duct. And then that duct merges with the urethra, and there's the prostate. and I'll talk about the bulbourethral gland in a second. And then the urethra, which could transport either urine or seminal fluid, but never at the same time. So I brought this up when I covered kidney physiology. We have a proximal urethral sphincter and a distal sphincter. Proximal and distal. How close is it to the bladder? Which of those sphincters do you have control of? The proximal or the distal? The distal. All right, it's going to be pelvic floor muscles. That means the proximal, you don't have control over that. Sympathetic and parasympathetic control. Urination is a parasympathetic event. It's a good time to go potty. So the proximal sphincter will relax and open up to allow the urine to pass. Ejaculation is a sympathetic event. And that is when the proximal sphincter clamps off, so that while seminal fluid's going through the transport tubes, you don't have urine going through the urethra because the urine would kill the sperm. Okay, so we've, believe it or not, covered that. We'll talk about the erectile tissues of the penis, the corpus cavernosa, sometimes it's called corpora cavernosa, and then the spongy tissue that actually surrounds the urethra, which is the corpus spongiosum. Do all those terms sound familiar to you, right? Okay, I'm going to keep track of the time here. Okay, so the seminal vesicles, we'll talk about it later, probably not today, produce an alkaline mucus. So does the prostate gland. It produces an alkaline mucus. Other than that, it doesn't really have much of a function. Why would alkaline mucus be important to be a part of seminal fluid? Yeah, to not, well, it would throw off the pH of the vagina because the vagina is acidic. And you want the vagina to stay acidic because of bacteria entering into it. The acid kills the bacteria. But I just said, if urine goes through the urethra at the same time that seminal fluid does, it'll kill the sperm. Urine is acidic. So the alkaline mucus in seminal fluid prevents the acid in the vagina from killing the sperm, protecting them for a long enough time till they get up the cervix and into the uterus. It's there to protect the sperm, because if there was not alkaline mucus in seminal fluid, the acidity of the vagina would kill them outright. They'd never make it very far. All right. Now, keeping track of the time here. Seminal fluid also includes fructose, a simple sugar. Sperm are microscopic little things, microscopic little swimmers. They have a long journey. Once they're deposited in the vagina, they have to make it through the cervix. They have to go up the uterus and they travel up the fallopian tube where most fertilization events take place. That's a huge journey for a microscopic cell with little swimming tail. So the sugar is there to provide sperm the energy to keep on swimming, keep on swimming. Dory, Finding Nemo? No, too old. Okay. Okay, and prostaglandin, seminal fluid contains prostaglandin. Prostaglandin causes the uterus to contract. Ladies, when you have your menstrual cycle and you get uterine cramps, that's your uterus producing its own prostaglandin, causing the cramping, contraction of the uterus to get that broken down menstrual fluid layer out of your uterus into your period flow, so that none of that tissue remains in the uterus where it can cause an infection if it remains. Prostaglandin causes uterine contraction. Why would it be good for seminal fluid to have prostaglandin? The uterus can contract and it might help move sperm up to the fallopian tubes where they could meet a possible egg. At least that's the going thought. The prostate gland produces a little bit of alkaline mucus, but you can live without a prostate gland. It doesn't really contribute much to seminal fluid. Now, the bulbourethral gland, it secretes a lubricant, a lubricating mucus. It produces its secretions during sexual arousal. It does not really contribute to seminal fluid as semen are going through the transport tubes. It produces a mucus that will go out the urethra and lubricate the head of the penis before sexual intercourse. Now, if there had been a previous ejaculatory event, there still could be semen in the urethra that was there before that could get flushed out to the head of the penis. Is it enough when the pre-cum, when you're playing around with pre-cum, could it cause a pregnancy? It's a low chance, but it's not a zero chance. All right, so be careful out there. All right, what else can I say? The prostate gland, and we'll get to this on Monday. Notice that the urethra goes through the prostate. So does part of the sperm transport tube, the ejaculatory duct goes through the prostate. There's this condition as men age where the prostate can slowly grow over time. It's just a natural process of aging. It's called benign prostate hyperplasia, or BPH. In some men, it'll cause no problem whatsoever. But in some men, when the prostate grows, it doesn't just grow out, it grows inward, and it can pinch that urethra. It can pinch the ejaculatory duct. So men with a bad case of BPH could become sterile because sperm can't quite get through that ejaculatory duct because it's being pinched. They also have a hard time emptying their bladder completely because this part of the urethra is being pinched. And you might have seen commercials on TV targeting men, of course, of a certain age, saying, are you tired of having to go to the bathroom repeatedly? You just can't go, or you can't empty your bladder. Basically, they're suffering from the prostate squeezing the urethra. There's drugs, there's surgery, but if they ever nick the urethra, you're in trouble. If they ever nick the ejaculatory duct, you're in trouble. And of course, if a tissue, remember the thymus hyperplasia, benign prostate hyperplasia, do you think those tissues, since they're growing a little bit abnormally, could become cancerous? Yes. So men of a certain age must start going for a regular prostate exam. Warning, future nurses. Some men are going to be highly resistant to this because there's only one way to check the prostate. You can check certain factors in the blood, but the only way to tell if the prostate is growing and is becoming a concerning type of growth is basically a lubricated glove going into the rectum, into the anterior wall of the rectum, and feeling the prostate, because it'll be right there. Nurses, you won't have to do this, I'm pretty sure. All right. But that's the way to check the palpate and its the prostate and its growth, is by palpating it. And men are not going to want to do this. All right. And I'm a big proponent of early detection, because if you detect something abnormal, you can get a biopsy and they send it out, and maybe it's fine. Maybe it continues to be harmless or benign. But if it's cancerous and you catch it early, you can save a life. But if you catch it late, then it hits the lymph system, which is not shown here, and then it's everywhere. All right. The men will be resistant to this idea. You have to kind of talk them into it and say, especially if they have a family history of prostate disease, like their brother, father, grandfather, you really need to get your prostate checked. And this is why. And it's really not that bad. It'll only take a moment. It's a little bit uncomfortable. Hey, ladies, we have to go for our cervix to be examined on a regular basis, because you don't want cervical cells to become abnormal. There's only one way to do it, is get in the stirrups, get that speculum in, and then get that little swab in there, right? None of it is what people want to go through. But you kind of have to talk to your patients. That's why you want to establish a rapport, so they feel comfortable with you, they trust you, they take your advice, and you might just save a life. Okay. So on Monday, we'll get into more detail about all this stuff, but that's a good enough job for Friday. Have a good weekend. It'll be nice.